Great, thanks, Philip. So my work is primarily with the RINGER program, and that's the acronym on the bottom left. It stands for Reforestation, Nursery and Genetic Resources, and this is a national program with state and private forestry, and I cover the 20 states of the eastern region. My office is at Purdue University, where I'm co-housed with the Hardwood Tree Improvement and Regeneration Center. So I'm here to give you some background on American elm, resistance breeding, and nurseries. So unfortunately, invasive species are a new and continual problem for our urban and natural forests. You may be acquainted with some of the most impactful invasive species shown in these images. Chestnut blade on the top left, which wiped out one of the most dominant trees of the eastern forests. Emerald ash borer, which is working its way through ash forests across the country. Butternut on the top right is an interesting one because we don't even know if the canker that's impacting it is native or introduced, but butternut is slowly disappearing from the landscape because of it. Conifers have not been exempt either. The hemlock woolly adelgid and white pine blister rust disease have been major pests. New to the landscape is spotted lanternfly, and one we are watching carefully. When these pests enter the urban forest, your choices for management are fairly limited, and they usually include spraying or injecting pesticides, and a lot of folks hope for biocontrol, but probably will not solve your immediate problem, leading to the most likely solution, which is to remove the tree and replace it with a different species. Ideally, you'd replace it with a cultivar or seed source that was bred for resistance, but very few trees with resistance to invasive pests are available to purchase. For this webinar, we'll focus on Dutch elm disease, which for many of you is no stranger to the urban forests. So how do we actually breed for resistance? Well, the first step is to identify candidate trees in natural forests that have favorable traits. We can't just create these in a lab. Secondly, we aim to conserve these selections by storing their seed, planting them into a common garden study, or making a graft or cutting of those candidate trees for a living clone bank. Later, if we have time and resources, we can make controlled crosses among top candidate trees. After one or more generations, genes for these favorable traits are stacked together. Selection is not limited to disease resistance. This photo on the right-hand side of the image shows a black walnut tree that was selected for its superior growth and form for the hardwood program here at Purdue. So once we get genetic material from these selections to a research facility, we can start to study the genetic extent of resistance. We do this with a common garden study, and it can take several different forms. On the left-hand image, you'll see young seedlings of eastern white pine in various stages of ill health. These were infected with the blister rust pathogen as one-year-old seedlings and monitored to figure out which one of these families had the best resistance. The planting on the right is one of northern red oak. These trees you can see are much bigger, and this plot is being used to evaluate stem form and growth traits to help us develop an improved seed source for timber production. These two studies look very different, but they share one important feature. The pedigree of each tree that you see in the rows and columns is known and tracked. This allows us to sort out genetic and environmental effects. Unfortunately, right now, we can only screen mature elm trees that are 5 to 7 years old, so the process for testing resistance to Dutch elm disease takes a lot of time. When all else fails and there is no natural resistance to a pest, we may decide to invest in a genetically modified organism as a means to save a species. A GMO, as it's called, is a novel genotype, and it's created by moving genes for desired traits between organisms that normally wouldn't be able to breed in nature. GMO trees are created in the laboratory, and they require decades of research to develop. And even if you're successful, the government tightly regulates the release of new transgenic, so there's no guarantee that a release will even be allowed. A GMO has been developed for American chestnut and is being reviewed right now by government regulators. I'm only bringing up GMOs mainly to remind you that American elm is not a prime candidate for a GMO. And this is mainly because we have cultivars of elm that are showing good resistance, suggesting that there may be enough genetic material for us to work with within the species. So I'd like to discuss some nursery jargon since it's relevant to this topic. And we're going to start with the term cultivar. A cultivar is a variety of a plant. It might be native, it might not be, and it's produced by selective breeding and usually sold as a clone. sometimes a cutting or a graft. So I have a picture here on the bottom right of a placard from a cultivar at Purdue campus just outside my office. And if you look at the label, you'll see a Latin name for the genus and species, Cerus canadensis. You'll see the cultivar name under it in single quotes, the common name, and the phylogenetic family to which it belongs. The image at the top right shows cultivars being grown in pots at the Forest Keeling Nursery in Missouri. Cultivars are usually grown and sold by private nurseries as opposed to state-run nurseries. The second term, it's less commonly known, is a nativar. And a nativar is a native plant variety selected for some horticultural trait. It might be flower color, stature, or something like that. Typically, they're sold as seed, used mainly for restoration. Some might be available for sale in urban markets. The placard on this Anis magnolia is similar to the redbud from the previous slide, except that it lacks a cultivar name. So the seed source may have been a nativar. It may have been some seed selected for some trait, but it may also have been a wild source. This aster bed, you can see on the top left picture, is at the Greenbelt Native Plant Nursery on Staten Island, and it was planted from a local, diverse seed source. They use this as a source of seed. They sell the seed or they sell the seedlings to folks who want to buy them. The beach plum, pictured on the right, is being grown at Pineland Nursery in New Jersey. The seed from this was collected from a wild area where previous collections had produced high-quality seedlings. So they returned to the same area to collect seed for production. Plant material that we use for wild or natural forests are usually not selected for a particular trait. The exception is improved sources that are selected for timber production. Wild seed sources, we just refer to them as seedlots. Sometimes we call them source-identified seedlots when the origin is closely tracked. So the bottom left picture is a serviceberry seedlot. It was collected and dried at the Missouri State Nursery. In the middle picture, you can see a tulip poplar growing at the Pineland Nursery, New Jersey. So they collect the seed from a known source and they germinate it, and then they grab the seedlings out of the bed and put them into pots. The label on the right-hand side is an example of a thorough source identification. The label contains the physical location of the seeds, the date collected, and this is at the Greenbelt Native Plant. So you may be wondering if source identification matters, especially in the urban context. And the answer is it depends. Generally in urban areas it doesn't, and the reason is most urban forests are populated with cultivars that are bred for or selected to tolerate urban conditions, that is high heat and anaerobic soil conditions. Urban trees are not expected to self-perpetuate, and that is an important distinction. The white oak pictured on the right was grown from natural seed, but if that tree died for some reason, Purdue would cut it down and replace it. We wouldn't expect it to repropagate itself. Now, it's different in natural forests, where our goal for reforestation or restoration is self-perpetuation. We expect the plants and trees that we establish to become naturalized. That means they need to reproduce on their own, which requires that they are synchronized with other plants and pollinators on that site. Lastly, we've been pretty lucky with elm because elm is a model species for resistance breeding and cultivar development, even though Dutch elm disease and it has other major pests. And the reason is elm can be grafted and propagated from cuttings fairly easily. Also, it produces copious amounts of seed from a young age, and the seed is something we call orthodox. That means it can be stored in the cooler for years, as opposed to recalcitrant trees like many oaks that cannot be stored in a cooler without killing them. Several cultivars of elm have been developed for urban markets, which is great, but we also want a population with improved resistance to plant and wild forests that will be self-perpetuating. One reason for this is that elm is a great species to plant in riparian areas that are now populated with green and black ash that may be decimated by the emerald ash borer. So with that, I will hand this off to Leila to continue.